Fermentation, the biotechnological platform for skin regeneration and aesthetics, but also in supporting health and longevity. There is a question that more and more scientists are asking today. Not how do we hide the signs of aging, but how do we stop the process itself? The answer comes from an unexpected place, a process billions of years old, a process used by nature to transform, amplify, and heal. Fermentation. Today I invite you on a journey at the frontier between science and nature, a journey that will completely change the way you understand skin, aging, and cosmetics. A molecule with a nearly century-long history, spermidine. It was 1926. British chemist Otto Rosenheim isolated for the first time a molecule from human and bovine semen. He called it spermidine. At the time, no one suspected that this biochemical curiosity would become, nearly a century later, one of the most studied anti-aging molecules in the world. Spermidine is not an exotic or synthetic substance. It is produced by our own body from the very first days of life. We find it in all living cells, bacteria, plants, animals, humans. It concentrates in wheat germ, fermented natto soybeans, mushrooms, aged cheeses, and, essential to what we discuss today, in fermented bee pollen. The problem? Spermidine levels decline progressively from the third decade of life, and this decline is not trivial. It correlates directly with the decline of cellular autophagy, the accumulation of senescent cells, and the acceleration of aging, both of the skin and the entire organism. The Nobel Prize for Cellular Cleansing. Autophagy, the year 2016. Yoshinori Osumi, a Japanese researcher, receives the Nobel Prize in Physiology or Medicine. The reason? He discovered and explained to the entire world the mechanism of autophagy. Auto, self, phagein, to eat, from ancient Greek. Autophagy is literally the process by which the cell eats itself, but in a profoundly positive sense. It captures its own damaged components, dysfunctional organelles, aggregated proteins, worn-out mitochondria, surrounds them, digests them, recycles what is useful. It is the cell's deep cleansing mechanism. But with aging, autophagy declines dramatically. In the skin, dysfunctional fibroblasts, degraded collagen, senescent cells that poison their surroundings. Systemically, accumulation of aggregated proteins, chronic inflammation, cognitive decline. The key question thus became, how do we activate autophagy naturally without drugs? The answer came in 2018, the study that changed everything. Science 2018. Frank Maddeo. Frank Maddeo and his team at Karl Franzens University in Graz, Austria, published in the journal Science, one of the world's most prestigious scientific publications, the global reference study, Spermidine in Health and Disease. The conclusion? Spermidine activates autophagy through a completely new mechanism, independent of classical pathways. It inhibits an enzyme called EP300, triggering a molecular cascade that initiates cellular cleansing. The results were remarkable. Spermidine supplementation extended lifespan in yeast, C. elegans worms, Drosophila flies, and mice by 10 to 25%. In humans, higher plasma levels correlated with reduced mortality and superior cognition. And the most bioavailable and concentrated natural source of spermidine? Bee pollen fermented through the SCOBY process, the biotechnological platform, fermentation, the fundamental transformation. But what is fermentation in this context exactly? It is not a simple preservation process. It is a biotechnological platform with three essential transformations. First, structural degradation. Fermentation breaks pollen's exin, one of the most stable biopolymers in nature, releasing the bioactive compounds trapped inside. It hydrolyzes collagen into small peptides capable of penetrating the skin. It solubilizes bioactive compounds from mushrooms. Second, generation of new metabolites. Fermentation does not preserve ingredients, it transforms them. It produces spermidine and its metabolites, microbial nanocellulose, pro-resolving epoxyoxylipins, bioactive peptides under 500 Daltons, soluble beta-glucans, and organic acids. Third, amplification of biological activity. Polyphenols gain increased bioavailability. Small peptides penetrate the epidermis directly. Postbiotics act synergistically, with superior efficacy compared to raw ingredients. This is the fundamental difference from conventional cosmetics. The paradigm shift. Conventional cosmetics has approached skin aging through a simple principle: substitution. We give the skin exogenous collagen, hyaluronic acid, antioxidants. The results are real but limited, because they do not address the deep biological causes of cutaneous decline. The fermented biotechnological formula does something completely different. It does not substitute. It reactivates the skin's own biological mechanisms. It acts from the barrier all the way to the nuclear level, with 14 integrated mechanisms of action, not 1 or 3, with documented senolytic and xenomorphic action, with autophagy activated by spermidine, with superior bioavailability through fermentation. This is not a better cream. This is a paradigm shift. The 4 biological kingdoms. The formula integrates active ingredients from 4 biological kingdoms. The animal kingdom. Polyfloral bee pollen fermented with SCOBY, the primary source of spermidine. Drone larvae fermented with SCOBY. Hydrolyzed bovine collagen fermented with SCOBY. And bovine colostrum fermented with kefir cultures. The plant kingdom. Fermented wheat germ, a source of vitamin E, zinc, selenium, and essential fatty acids. The fungal kingdom. Tremella fusiformis, with polysaccharides for deep hydration. Agaricus bisporus, a source of spermidine and ergothioneine. Saccharomyces yeast with soluble beta-glucans. Bacterial microorganisms, SCOBY and kefir cultures, which endogenously generate an entire universe of bioactive compounds. Polyamines, polysaccharides, microbial nanocellulose, organic acids, bioactive peptides, exosomes, bioactive lipids, vitamins. 4 kingdoms, 7 active fermented ingredients, 2 fermentation types, 14 mechanisms of action. Stay close. Part 2 is coming and takes everything to the next level. Don't forget to like, subscribe, and hit the notification bell so you don't miss what's next.